So good afternoon, everyone. So I'm going to be passing you through the extraction of the TB DNA using CTAB method. So why do we need to extract the DNA? So we need the DNA to, we need to get the DNA from the bacterium, which are going to use and put with barcode for the sequencing thing. So that's the reason why we need to extract the DNA. We use it as a template. So DNA requires, sequencing requires good DNA, and you need to get good quality DNA, which has no impurities and contamination. So for impurities, we get impurities from the detergents that we're going to be using. So we need to make sure that we do the washing properly so that we don't leave any leftovers. Then for the quality, we need, for the kit that we're using now, we need 50 nanograms per microliter. But we're going as far as 400 nanograms per microliter because we just don't want to get smaller qualities. Then for high molecular weight, we need long reads. So we're going to be as gentle as we can so that we get long strains of DNA, because we don't want short strains. Like when sequencing, we need longer strains. Then DNA extraction options, the number of commercial kits that we have the Caren kits, we have Zymo that we use for extractions, but those are not good enough for extracting TB. Yeah, so we have, I think, I've not tried extracting DNA. I've not tried extracting MTB using the kits, but I don't know how good they are.
I think generally you find they're not powerful enough to break down so you probably will get some DNA, but somewhere between 50 and 400 nanograms of DNA, and that can be quite difficult to achieve with the commercial kits. So this is why I think CTAB is generally used.
So why DNA is difficult to extract? It has a thin, complicated cell wall. It has, I think it's made up, 60% of the bacterium is made up of lipids. So we have to go through the lipids to get to the DNA. So we have to break it. So for the kits, I think that would be difficult, but then because we're doing CTAB and we're using SDS, that would be easy to break down the cell wall and the lipids. So to extract DNA, we have to balance between opening up enough DNA and getting longer reads. So that's why we have to be gentle enough, to break the bands, the strands, and not to break the strains and get quite good quality DNA. Yeah, so you have to be gentle and rough at the same time, but not very gentle again. Yeah. So this is why it's difficult, because of lipid layer. So you have to get through the lipids. So use detergents such the CTAB detergents. That's one you use to break through the lipids. Then we use SDS as well. Oh, if you're wondering, CTAB is cetyl trimethylammonium bromide. Yeah, so that's the detergent we're using to break down the macrolic acids and the lipids to get to the DNA, just to disrupt it. And the safety requirements, TB is a hazard tree group organism, so it should be processed in an appropriate laboratory. And it should be unless it's validated. So for our case, we did the killing at Mr. Solo's lab, the TB lab. We did the killing from there, the heat killing. Then from here, we just did the chemical lysing and sonication. Then for killing, we're doing it at 85 degrees. So it's, people are still, the literature says, some say 80% is the best, some say 95% is the best. So for ours, we validated that 80% is the best, like 80 degrees Celsius is the best for 30 minutes, the 20 or 30.
I think it depends. Basically, it's in some labs, certainly in the UK, our lab has validated that 80 degrees for 30 or 20 minutes isn't enough to guarantee that you killed all of the bacteria. And so we would recommend 95 degrees, but the only trouble with that is that the higher the heat, the more likely you are to start denaturing the DNA. So with long read technology, you really want to have that long read. So the way we do it is we have it 80 degrees for 30 minutes, which should in theory kill the majority of the bacteria. But then we do all of our CTAB processing up to the isoamyl step in our cabinet, because until that point, we have essentially said we don't believe that absolutely every bacteria will have died at this point. But I know that the UK has very strict Cat3 regulations or BSL3 regulations compared to the rest of the world. So if you in your laboratory have validated that basically whatever you have validated it for, that's fine. That's your own, obviously, validation and your own protocols and SOPs. But we would recommend to keep your DNA as intact as possible without sort of disrupting it. 80 degrees is definitely better, but just consider where you're going to continue to do the rest of your CTAB method. As I say, I think a lot of you have already done or are already doing CTAB, so it's up to you how you do it, but it has to be validated.
Yeah. So for us, we're hitting at 80 degrees Celsius for 30 minutes, which is high enough and gentle enough not to break the DNA. Then sonication, chemical lysis, and precipitation. So for the heat killing, debate has been there on which one is better between lower temperatures to protect the DNA or higher temperatures to ensure you're killing the bacteria. So for us, we went with the lower temperature, which is 80 degrees Celsius, but for a longer period, because I feel that if we hit at a lower temperature for a long time, that would be long enough to kill the DNA and not to break it. Because if you use the higher temperature 95, some will go as high as 100. That would denature the DNA. So things to consider to get effective, to get high molecular DNA. So type of sample you're heat killing. So it depends on the type of sample you're heat killing. If it's a sputum sample or an isolate from a colony from the culture. So for us, we're using the colonies from the cultures. We're not using direct sputum samples. And the concentration of the sample, how many bacteria are there, and the volume of the sample. So we got, after adding the lysis buffer, we got to 400. So our starting volume was 400. So water block versus heat, water bath versus heat blocks. So for me, I feel using a heat block denatures the DNA. For some, we have an epindorf, and some we have bacteria up there and down there, and the heating block usually just gets very hot on the downside and not the upper side. So some DNA won't, like the temperature won't get at 80 when you put into the heating block at the top of the epindorf tube. But for a water bath, you get equal temperatures. And I'm not okay with the heating block, so I go with the water bath.
I think it's just a case of validating that your thing is definitely killing everything.
Yeah. So sanitation. So basically what we're doing is we're disrupting the cell wall to get enough DNA to come out of the cell using sonication. Yeah, we do this for 15 minutes, which is a very long time. Then the chemical lysis, so that the chemical lysis, we start with lysosome that breaks down the cell wall. We use about 50 microliters of lysosome to our 400 microliters of the solution where the colonies are. So from there, we put the protein escape that breaks down the protein lysis and releases nucleic acid into the surroundings. Then we use CTAB. How much percentage we use of CTAB?
It's not 200. I think it depends on the process, but I think we use 200.
No, we use 100 microliters of CTAB to denature the cell wall and the proteins. And then for SDS, so the SDS stabilizes the membrane and the lipids. It's like just after it helps, so the SDS helps the protein escape, like just right after the protein escape breaks down the cell. The SDS helps to carry on with the cell destruction. Then for DNA precipitation, DNA is hydrophilic, as we all know, it dissolves in water but not alcohol. So that's why we elute in water. For us, we're eluting in water, just nucleus-free water. So to separate the DNA from the cells, alcohols and salts are used. So in this case, we're using ethanol and isopropanoate. Yeah. We're using alcohol, ethanol, and isopropanol. Yeah, to precipitate the DNA. So organic solvents are used to separate DNA to an aqueous phase, leaving lipids and proteins in the organic phase. So what happens is, if we add the alcohol, which precipitates the DNA and leaves the other things that we don't want insoluble, which we wash off. Then so that's the washing step. This is the washing step using ethanol and isopropanol. So when you add isopropanol, also precipitates the DNA to remove the excess salts and the proteins using alcohol, which we wash off. Then isopropanol is used in reference to ethanol, as low volume for precipitation can be used. So in a ratio, so it depends on the amount of DNA that you add, the amount of DNA that you started with. That will, like, you have to know the amount of DNA that you start with in order to know the amount of isopropanol you're adding to the sample. So if you started with a volume of 150, you have to add isopropanol of 150. If you started with a volume of 200, because you're having different volumes of DNA from the colonies, so that will, you can't just add, like, you're adding 1 to 1. So 1 to 1 will depend on the sample volume that you have. Then for the yeast, it's used during the alcohol precipitation to effective precipitation and aid in the recovery of small amount of nucleic acid and improve the DNA yield. Then DNA precipitation, we add the salts and alcohol, then we centrifuge to get the pellets and remove the supernatants and resuscitate in water. So explain more. So the precipitation, am I on the same slide?
Precipitation, I think it just represents each step.
Yeah, it's just each step, yeah. Okay, so that's, if you go back to the previous one. So at the beginning, you've got your DNA in pre-suspension, right? Then because we've added the alcohol causes the DNA to precipitate, right? So it comes out of solution. So then when you spin, you get all the DNA collecting at the bottom of your little epineural tube, right? And then you form a pellet at the bottom of the tube. And then you just take off the whatever fluid, this fluid on the top. You just get that out, right? And then you're left with the pellets, then you add water, and then the DNA goes back into solution. That's
It's
basically.
really important as well to allow it to completely dry out, and so you don't have any salt or alcohol there. I've been speaking to, we've got a team at Nanopore who we work with, and they were saying that with TB, or it might not be necessarily TB, but with the CTAB extraction method, what we're finding, and we found this as well, is that when you sequence TB and you compare it to, say, a Klebsiella, you find that it doesn't sequence anywhere near as well, and that's why traditionally we put a lot less TB samples on per run. But what the ONT team are saying is that they think there's something that's co-precipitating, either because of CTAB or because it's TB, so maybe something from the mycolic acid layer that is interfering and interrupting and stopping the transposase from binding to the DNA and allowing the barcodes to stick. So if the barcodes aren't sticking to the DNA, then the DNA isn't going to be going through. If it's not able to stick anything to the DNA, then it's not going to be actually sequenced. So it's there, but it's not processed, so it can't be sequenced. So we're currently working on a modified CTAB method. So the CTAB bit is there, but then instead of doing ethanol precipitation, which is obviously leaving something in there with it, there's a sort of a spin column step instead, and so it's purifying it in a slightly different way. We're currently working on that at the moment, so hopefully in the next few months we'll be able to say whether that really works or not. But at the moment, they're definitely, it doesn't affect the ligation kit for Nanopore, so it's definitely something to do with the transposase that actually, as I say, binds the barcodes to the DNA, we think. So yes, CTAB, excellent, but can be improved.
Yeah, so to get rid of the ethanol and isopropanol, we dried for 24 hours. Yeah, to get rid of everything. Then DNA can be stored at 4 degrees. So we stored ours at 20. So when we're done precipitating up the DNA and everything, we leave it overnight in order to evaporate the isopropanol and the ethanol. Then we add water to reconstitute it and leave it at 4 degrees Celsius for a day, right? We left it for a day.
Yeah.
But again, the other time that, remember, we did 2 groups of.
Yes.
So the one that I did for 2 hours was good, like came out good. And the one that we did overnight didn't come out good. So I don't know why. We can look at that. Yeah.
You've got to allow enough time to allow the DNA to reconstitute back into the liquid. So either you can leave it for, say, 65 degrees for an hour, for instance, or you can leave it overnight in the fridge, and it will be slower to go back into the water. If you, for instance, cubited it immediately, you would find that you had, try saying that. You will find probably that you would get a lower reading than if you left it for 24 hours, because obviously your DNA is still probably stuck on the bottom, because that pellet, remember, you dried to a crisp, and also it's been centrifuged, so it's going to take quite a lot of effort to reconstitute that pellet. So it's always worth, when you've done CTAB, if you don't already, leaving it for 24 hours or as long as possible, and then cubiting it or doing your quantification analysis, because it probably won't be accurate immediately afterwards. That's not the same if you were doing a spin column based one, because it works in a different way, and you don't have that solid little pellet at the bottom.
Yeah, I think this is it. I think this is what we need to get good quality DNA for sequencing, so that we just don't put in rubbish, as Lindsay says, and get out rubbish when we're sequencing. Yep.
I think what we have found certainly is that the CTAB or the extraction method for whatever DNA you're doing, TB, BCG, E. coli, whatever, it's really, really important to get good quality and good quantity, because as you'll see when we do DNA quality control part this afternoon, if you put rubbish DNA and that's all sort of smeared and it's not a nice neat band and it's not really high quality, then you're not going to sequence very well at all. It's going to be it's just going to be rubbish basically. And also one sample that's really poor can affect the sequencing sometimes of the other samples. So it's worth your effort to repeat the extraction rather than worth your costs to repeat the sequencing, because obviously it's not cheap to do that. What we put in the protocol is our kind of modified version of CTAB. You might not all do all of the things that we put in here, but our clinical trials team, we do quite a lot of sequencing, and our clinical trials teams have modified it, so adding the yeast tRNA to help precipitate out like all the final bits of DNA. This version of our protocol is kind of what we so far have as an optimal version for sequencing. So you might use something different, but that maybe because you're not using it for sequencing. Sequencing is, as I say, you need a lot of DNA, you also need good sort of quality DNA. If you're doing PCR, you don't need that much, and so it doesn't really matter if you kind of smash it about. So yeah, as Caren was saying, you have to be very gentle with it. Don't pipette it violently up and down because you're going to shear all that DNA. You have to treat it like a baby and make sure it's very happy and very long. So yeah, as I say, we're not going to do CTAB as part of this, but if you want to talk about CTAB and how best to kind of optimize it, have a look through the methods and see if it's different, and we can, as I say, talk about kind of maybe trying to optimize it. And hopefully in the next few weeks and months, we might have this new optimized ONT version of the protocol, which might work even better and hopefully eliminate whatever it is that seems to be inhibiting TB or inhibiting whatever is done with CTAB. And then maybe we should be able to put more samples on per run and hopefully make it cheaper as well. So yeah. Does anybody have any questions? I think a lot of you do CTAB anyway, don't you?
The ones from the TV lab.
Yeah.
Is that enough? Do you use tRNA? I don't think it's a commonly used thing, like to co-precipitate. Yeah.
Yeah, we, no, I don't really. For cell sequencing.
Yeah.
Okay.
Quite similar. It's just, I don't know, when you use the money, instead of water, you can make the last.
So that's another thing. We found that with the best one in the world, you don't get huge amounts of DNA from TB because, as Caren was saying, you've essentially got to smash it with all of these fairly violent, sodium sulfate and all of that detergents, but also try and maintain really nice clean DNA. And it's a real balance. So you are inevitably going to get less DNA because you're trying to be gentle with it. And so I've been eluting into as little as say 20 microliters, and obviously that's a really tiny amount, whereas usually you would elute into a bit more. So for sequencing, to get the right amount of DNA and a good quality DNA, you kind of have to tweak what you're doing, what you might be doing already to get what you need, because it's different to what you might need for say PCR or hay or those kind of things. But yes, rubbish in, rubbish out. Remember that. But yeah, we can do the quality control stuff this afternoon on the samples that we've got. And hopefully you'll see what I mean. So
I heard you mentioned that we're trying to the protocol that you're trying to develop that combined CTAB with stream column methods. Have you're yet to publish it or?
so it's something that Nanopore have been developing. I
Okay.
can't talk too much about it because it's still in
It's still in
development.
development, okay.
We are the first people that they have given the protocol to test that. I did some extractions before I came out. So we have very strict rules about setting stuff up in the Cat3 lab. So we tested it on recently, and then an abscessus sample from abscessus samples, sort of at Cat2 level. So we're going to test them against some TB that we've got from our original CTAB version, and then also against some E. coli as well, because if you run this kind of CTAB extracted TB against something that's been done in a spin column kit like E. coli, I suspect it might be a combination of E. coli is just much easier to do, and also spin columns I think possibly purify whatever this thing is out better. But when I ran an E. coli against the TB, you couldn't even tell that I'd run TB. I got so few reads, it was really pathetic. So what I'm going to do when I get back is test our new version of the CTAB, the BCG and the abscessus against an original version of TB, and then sort of just an E. coli or Klebsiella that we've got in our sort of collection that we've done before, and see how well the new version compares to the original. So yes, it will be, if this works, then as I say, we're the next lab for them to have tested. So we're trying to work out whether we can actually get what they are seeing in the first place. So they think it works on BCG, but we're now the ones testing out. So watch this space. Hopefully, fingers crossed it will work, because if we can get it to work in the same way that E. coli does, as I say, you can start to run maybe 24 or even more samples on each flow cell. We've done quite a lot of ground negative sequencing with various student projects, and we worked out that with the, the whole way through doing the extraction kit and then doing the sequencing preparation and the flow cell, if you're putting, say, 24 samples on, the cost of it actually comes down to quite reasonable. It's something like $40 to $60 per whole genome. So yes, if we can get this extraction technique to work better, then we might be able to get it down as well, which is obviously again something that's really important, because people aren't going to want to run it and roll it out if it's prohibitively expensive. So as I say, this workshop and this protocol is very much, it's not the literal best, but it's very much a, can we do it cheaply, can we get good accurate results, and most importantly, can people who aren't complete experts on sequencing and bioinformatics do it as part of routine stuff? Because not everybody is obviously going to, you might be a TB routine diagnostic person, you might be, an epidemiologist. It's, not everybody is going to have bioinformatics skills. And what hopefully we'll be able to teach you on Friday is the very sort of basic intro, so you can go from those files all the way through to having, this is resistant to this, this, and this, and it's lineage this. So yeah, that's the aim anyway.
Then there's another question was with regards to the original CTAB network. How do you dry your pellet? You embed it, you just open it, and then expose it. How do you dry it?
So we open the lid, then we dry it on the heating block, but I didn't. I did 3 batches. So the first batch I did it on heating block. The second one was in the hood. Yeah, so I think the ones that were on the heating block did good, and the ones that were in the hood. So you just open them up and leave them to dry off.
You have to try and compress off the most amount of ethanol, but at the same time, often trying to see the pellet.
Trying to see the pellet.
I mean, you've got to have very good eyesight. So you don't want to suck up with pellets. So
Exactly.
it's kind of, again, it's a balance. So if in doubt, I would leave it in, and then it will just take much longer to evaporate.
Yeah, overnight.
If your room is warm as well, then that helps. But if your room is air conditioned like this one, then it will probably take a bit longer because it's cold.